So thank you very much. My background is purely biochemistry, and actually I even mostly studied and worked in medical environments, so it's gonna change a bit the tone, I think. But I mean, in my conferences and so on, I'm usually considered like the weirdo that does very fundamental things. So in this audience, I feel like on the other side of the scale. But in any case, so I hope I can give you some interesting information about a process that we talked already about a lot here, metabolism. And I will not have a lot of I mean, I want to kind of convince you that there is still at the basis of establishing even how metabolism works. I mean, all the reactions that constitute metabolism are far from being all figured out. So you are already working on emerging principles and universal rules that that are maybe behind all the regulation of metabolism and so on. But there are some poor biochemists that still kind of try to work out actually what are all the reactions that make up our metabolism. And so you will, I think, over the presentation, you will start understanding the title. But it's important for me before starting the science to give a big thank you to our organizer for inviting me here and for giving me this opportunity to present. And this was yesterday evening, and I think he always only feels good at a place where there's some sea once he has caught one of those. So I wanted to share this with all of you. And so yes, coming back to metabolism. It is already a very complex network. It was shown, I mean, different pictures of this were already shown, but actually, I think, and I know many of you like to not at all consider all this complexity because they are already so annoying. So you way prefer A to B, B to C, etc. But basically for us, there's still actually a lot of things that are not in this map. And that's what I'm going to show you, what the kind of things that are still missing. But just to start, I mean, I wasn't sure that we would get such an elaborate introduction already to this whole process, but as Matthias said, repetition is important for learning. So I'll give a very few slides again about just the general principles, you know, how we can boil this down, and if you want to understand just what's underlying all of this complexity. In the end, yes, we are basically breaking down complex metabolites. So metabolites, you remember, it's the small molecules, the small chemicals in our organisms, in our cells, that can be, however, bigger or smaller. So what we take in from nutrition, there is a lot of more complex and bigger molecules that we need to break down through pathways. So pathways are these reaction chains that in that case we call catabolic reactions to bring them to more simple products that we can then use as building bricks, let's say, for doing anabolism, the other pathways that build more complex molecules that our cells, our organism can use to grow and to repair and to maintain. And while we are doing this catabolism, we are also extracting energy and putting it under a form that we can use, namely ATP. And as you remember also, we phosphorylate ADP to ATP, which is a free energy source, and we reduce NADP, a very central cofactor, to NADPH, which both then are used in this anabolic or biosynthetic reactions. So energy, at least one thing that one of the terms of the school I can definitely name here that is also central to metabolism is the energy metabolism part. So also here you saw the pathways. I show them again. So if you want to summarize like which are the main pathways for energy metabolism, classically, one shows the glycolysis the TCA cycle from glycolysis. So glycolysis is this oxidation of glucose down a 10-step pathway that leads to pyruvate, which is then converted in the mitochondria to acetyl-CoA, and which then enters the TCA cycle, which is in the mitochondria, then leading to the mitochondrial respiratory chain, which is in the end also producing quite a lot of ATP. And from glycolysis, you can deviate one of the intermediates, which is a term that Matthias also used. So these metabolites that are on the path here, going down the glucose oxidation. The first one actually that you derive from glucose, glucose-6-phosphate, enters a pathway that is called the pentose phosphate pathway, where there is a reductive, so a part where you actually generate a lot of the NADPH that we are then using in synthetic processes, but also in protective processes against oxidative species. But we also generate ribose 5-phosphate, which is an important building block for our DNA and RNA. And then here on the side, another important energy source, of course, are our lipids, which we degrade to fatty acids, and for catabolism they enter under an activated form into the mitochondria, and there happens a pathway that is known under the name of beta oxidation, and we get a lot more in terms of yield. We get a lot of more ATP from fatty acids than from glucose actually. So glycolysis, I said the gluconeogenesis is actually a pathway that basically, I don't want to say the reverse pathway because there are different reactions involved, but it goes, instead of going down from glucose to pyruvate, it goes up from pure pyruvate or some other incoming sources to glucose. So that's also a difference maybe to what we have, how we have considered metabolism. I'm more talking about human metabolism, where we have a different organization. So where you have a high specialization of the different cell types, of the different organs, also in terms of metabolism. And so you will see, I have one slide on this, that in different organs have different metabolic roles, and you have very different types of, even the map is different of the metabolism, and the regulations are different. And so some organs, for example, do this gluconeogenesis a lot because they are kind of sources for giving glucose to the organism in times where we don't have any more in the blood from nutrition. And others don't do it at all because they have not this role, like muscles, for example. So glycogen is our also another source of glucose when we don't get it directly from food. That's the storage molecule under which we store glucose, and can, during the night, for example, go on like fueling our brain with, so that it can continue to do the essential functions. Although we are not eating, and otherwise, I mean, without the glycogen, the glucose levels would fall below what we need to maintain our essential functions. Then the TCA cycle, we talked a lot about it. Oxidative phosphorylation is what then follows after, where the NADH is channeling its electrons into in the respiratory chain. Pentose phosphate pathway, I described fatty acid degradation and synthesis. And amino acid degradation and synthesis is actually not represented here, I think, but this is what happens at the stage where you have really eliminated all of your resources, glycogen, all your fat is burnt up. Then you get into an extreme state of starvation, and you start breaking down your proteins actually, and then you can also use amino acids as a source of energy. Then also clearly a notion that we talked about, very important, this metabolism is heavily regulated. So, there was this nice representation with a bow tie that goes to a knot. So here we are in the knot, and it's in these pathways that you have a lot of regulation. I'm careful with these terms here, but of course all of the metabolic reactions are reversible, but some are more than others, and it's typically at the reactions that are far from equilibrium that a lot of regulation is happening. Also, another kind of recurring theme is that we regulate at the many, I mean, not only, and I think Matthias showed some other examples, but often pathways are regulated right at the first branching point, because then, you know, you don't waste resources to go into these pathways and build molecules that are not needed at this stage. So you kind of choke them off right at the start. But of course, there is also further down the path regulation going on. And here in blue, I'm showing you know, some of these main regulated points in some of these pathways, which are quite a lot studies, and we talked already about phosphofructokinase. And I think maybe Alex is going to talk about calcium regulation here in the TCA cycle of some of these enzymes. So you will hear more about it. And here is what I said, like with this in animals, in humans, for instance. So there is a specialization of metabolism in the different organs. So the communication between them to adjust the metabolism is done via hormones. Sorry. Oh, going back. Via hormones, certainly you have heard of molecules like glucagon or insulin, which are very famous hormones that regulate metabolism upon taking a meal or upon entering into starvation. They do this by many different mechanisms, triggering cascades, for example, of phosphorylation, protein phosphorylation, where you activate, deactivate enzymes, bringing receptors to the membrane that will then make that nutrients can enter into the cells. And different organs have, you know, react very differently to this glucagon and insulin, and because they have different metabolic roles. So in the case of, I'm just stating a few of them. So liver is actually what we would call our glucose sensor. So it's making sure that we keep this glucose homeostasis. I mean, we say usually that we keep our glucose around 5 millimolar in the blood, and glucose is mainly watching out for that. So when you are taking a meal, it's the liver that senses this. And I'm not sure if it's what we are all saying. Maybe it's actually the flux that it senses, but what liver has different from other organs is this first enzyme of glycolysis, that is a phosphorylation of glucose into glucose 6-phosphate. That enzyme in the liver, it's a form of the enzyme that has a that's actually slow at activating once there is glucose. So it has a high Km, meaning that normally it's working relatively slow. But then once glucose concentration is going up, it starts picking up speed, and that's why, you know, why it's believed that the liver, I mean, that's one of the things that contribute that liver is this glucose sensor. The muscles are, as you can imagine, mainly breaking down glucose for fueling the energy that's needed for contraction, for instance. And in the muscle there, so also the liver, because it has this function of keeping the homeostasis, so it reacts to high glucose by starting doing glycolysis faster, putting things into storage, into glycogen and so on. But it's also sensing when there's low glucose, and in that case it starts degrading glycogen or doing glucose from other sources via the gluconeogenesis. While the muscle, for example, is not doing that. It's just like breaking down glucose and extracting the energy to execute its functions. The brain, we said already, is highly reliant on glucose to function. It's a big glucose consumer in the organism, and that's also why it's so important that liver keeps this glucose homeostasis. And also specificity of the brain is like it's not breaking down fats, so it's using glucose, and once this is really gone and we have exhausted all of our storages, etc., then liver starts to break down fats into something that we call ketone bodies, and that's then brought to the brain for further fueling all of the reactions that the brain needs. Okay, so I think. And so, as I told you, this map is already like thousands of reactions. As you remember, all of the arrows represent chemical reactions, basically conversions of one metabolite into another one. And a big majority of them are catalyzed by these proteins that are called enzymes. And while there's already a lot known, and as you have heard, it's actually one of the oldest biochemical processes that has to be studied for more than 100 years, we elaborate this map. It's actually still not complete, and I think there is still a lot of gaps to fill and actually extensions to do on this. And one indication for that came actually with what we call the genome revolution or genomic revolution. So, as you know, in biology, like a big push has come with the acceleration of how fast we can sequence our DNA, just experimentally, technically, with next generation sequencing. And so around in the 90s and beginning 2000s, we became able to sequence entire genomes. So really decode it and start actually understanding in its globality, like how many genes are actually there, and what is the space in which we are playing in terms of the whole proteome, the whole transcriptome, etc. And so that turned out for a species that we talked already quite a lot about, budding yeast. So we knew at that stage, actually this is now, I saw, down to 6,000, so there have been some corrections done. Probably at the time where we wrote this review, there was still some redundancy and some misannotations, but there is around 6,000 proteins that are encoded by the whole genome in budding yeast. And there are around 20,000 of these proteins that are encoded by humans. So that's the human genome. So that's something that we knew then. But a surprise was that when then people started looking into all these sequences, all these proteins, turned out that for a reasonable proportion of them, we had yet no clue with everything that was done until then, with classical genetics that usually worked by, okay, we have this yeast strain that is growing weird, so now we want to understand why, and so there were a lot of experiments done to pinpoint in the end the gene that was in this black box of a genome that we didn't know it in its entirety. Well, now we know the whole genome, and we can look at all its parts, and we see that actually by these classical genetic efforts, we have figured out maybe 60, if maybe a bit more than half of the, or let's say 2/3, for depending on the organism, of course, of the functions that it actually encodes in terms of proteins. So that's only to even talk about the coding part of the genome, because the coding part is something like, so the coding part with the genes and which lead to the formation of the proteins, I think is something mind-blowing, around only 2%. So the rest is all non-coding DNA, which is even a bigger box to all still disentangle. But let's focus here on the coding part. We know 20,000 genes in humans, 20,000 protein coding genes in yeast. But if you go into databases and look at their functional annotations, you remember proteins, it's not only enzymes, it can have a lot of, yes? Yes, exactly, that's what I want. I'm talking here globally. So proteins can be enzymes, of course, we have a tendency to here equate it to that, but actually, you know, proteins do a lot of other things, transporters, which are also contributing to metabolism, receptors, antibodies, transcription factors, like so many different things, small peptides like hormones and so on. But so here I'm talking about all the functions, but if you go into protein databases for each type of protein, you will have, you know, it will say more or less precisely what it is doing. And if you go there and then you look at these annotations, you see that about 1/3 you have these terms of putative, uncharacterized, unknown, domain containing, you know, this kind of annotations, where actually it means basically that we don't know yet what they are doing. And that it's at that time, it was in 2017 when we did this, when we looked into this to try to get a grasp a bit on that. About a third, as well in yeast and then in humans, about a third of the proteins had no well-established function yet. And because of our interest in metabolism, we were curious to figure out more or less, get an estimate of how many of these proteins are actually enzymes, which of course we cannot know for sure because by definition, as I said, their function was not yet elucidated. But we can use some tricks, like in a way from evolution, to try to get we try to use these tricks to estimate more or less, okay, based on sequence homology basically, like how many of these proteins looked like proteins, just based on their sequence. And I will not go into the details of this, but basically we went into the databases, took all of the sequences that at some point in some organism had been associated with an enzymatic function. And I will very briefly explain you what this EC number is. So in databases, that's what you find as a linked to enzymatic proteins is this EC number. So we took all these sequences, made for each EC number, so each enzymatic activity that had been described in any kind of organism, like across organisms, made for the similar sequences alignments to find out the conserved parts, based on which we could build some models, then scan them against the proteins of our interest, so the humans and the yeast, just because we are working on them mostly, to then find out like how many of these proteins in this proteome kind of look like enzymes. So indeed, then they were associated. Of course, by this way, we found all the known enzymes already. So they had a full EC number. That's when the full function is known. Then these four numbers are all given. But if they have no EC number associated, of course, it's still totally open what it could be, but they could also have incomplete EC numbers, which means that basically the precise enzymatic function was not figured out. I'm just giving you that information because maybe your work in database can be interesting. And here, as we talk about enzymes, it also allows me to give a bit more chemical background. So the enzymes are, we divide them into 6 major classes based on the chemical reactions that they catalyze. These are oxidoreduction reactions, so that's class number 1. All of the enzymes that do this are in class number 1. The transferases, the hydrolases for the hydrolytic reaction in digestive processes, for example, lyases, that eliminate groups of chemical groups and create double bonds, isomerases that kind of interconvert some pieces, so for example glucose 6-phosphate into fructose 6-phosphate in glycolysis, and ligases that are actually consuming energy to form new bonds. So these are the 6 big enzyme classes, and based on this I can now explain you this classification of the enzymes that we do. So EC just stands for enzyme commission, then this first number gives you the major class. So is it an oxidoreductase, a transferase, a lyase? So that can go from 1 to 6. The second number, then we refine further into the enzymatic reaction, so it gives you the subclass. In this case, if that's a 1, it means that it's an oxidoreductase that works on a CHOH group. If then in the third, then we go in the subclass. In the third place, we also have 1. It means that it's an oxidoreductase that works on this kind of bond and that uses NAD or NADP as a cofactor. And then the last number is actually just a serial number. It has no meaning. It's just like, the first one that we discovered was, or that we put into this category was alcohol oxidoreductase, and we will just like call it the number 1. And but this, for instance, this number 1, or even this 111 enzymes of those, there are already more than 400 different ones known. So this number can go from 1 to 400. showing you already, and that's maybe something that you can remember. I told you NAD, NADP, these are very central, not really metabolites because they are not, well, actually, yes, they are also used per se, but often they act as cofactors of enzymes, so they are basically just cycling between the NAD and NADH form. But there are many, many enzymatic reactions in metabolism that use NAD or NADP. Yep. When I see this, I know, yes. Then because then the full reaction is well, except if yeah. Yes, it should. I mean, typically if there would be still, you have these linear pathways. If there is one reaction that we are still missing, and we see that, it's actually going from a non-phosphorylated metabolite to a phosphorylated one, and then we find an enzyme that maybe we characterize isolated in vitro, and we start figuring out it's actually transferring a phosphoryl group, and, what is its specificity, then we will be able to fit it in. If this fits in a known pathway. I mean, of course, there is still even entire pathways that we elucidate, but if it's a gap, then yes, it can give you this hint. And so now, when we applied different strategies to try to estimate only how many of these proteins are actually maybe enzymes of unknown functions, so not proteins, not just proteins, but enzymes of unknown function, it turned out that we remain with this 1/3, and 1/3 of the total proteins are proteins of unknown functions, and again, it seems like more or less 1/3 or even more of these. Depending on the organisms, again, of these proteins of unknown functions are enzymes of unknown function. So this gave us a little bit also, a raison d'être, as biochemists who want to still discover enzyme function. It really shows that there is still a lot of work to do, and it also shows you that, manifestly, the maps cannot be complete. Although you could say, well, enzymes also, I want to make this comment, are not only working in this intermediary or primary metabolism. They also, what I'm stating here, it's basically proteins with a catalytic function, which does not mean necessarily that it works in metabolism. So, metabolism as we understand it. It can also be enzymes that put a phosphoryl group onto a protein, which in a way it's regulating metabolism, but can also be involved in other functions. Or an enzyme that is modifying a tRNA in a certain way. So this is not what these are not the metabolic. These are not only metabolic enzymes. Can also be other type of enzymes. Yes. Yeah, so you can of course now with what we call reverse, you can find it out because we have all these techniques of just knocking out genes. So now that we know on which kind of genes or enzymes to focus to, because there seems to be unknown function, we can go and knock them out in a genome of our species of interest, and then see whether it affects growth or whether now all of a sudden the cell cannot grow on a certain substrate, etc. So that would be one way to figure out whether it's really having a function. However, I have to say that within these enzymes that we still don't know what they are doing, typically they are phenotypically rather silent. So these obvious phenotypes that you saw a lot, in the first genes, etc., that were identified, they draw our attention, because if you indeed have a mutation in them, really something catastrophic happens, and the cell is not able even anymore to divide. Typically these, and that's probably why we are lagging behind into discovering their functions, they don't have such a dramatic, I generalize, right? But many of those are phenotypically, they don't give such an obvious thing. But then if you go into molecular characterizations of your knockout of these cells, for example. Then you start maybe seeing differences at, if you have these techniques, the omics techniques that allow you to analyze, all of the RNAs that are for the moment expressed, or all of the proteins that are for the moment in the cell, or all of the metabolites, and then we talk about metabolomics. So you might see differences that then hint you towards that there is indeed a function. But yes. Yes.
This might be a silly question, but I noticed that, like, for the proteins in general of unknown function, there's like a very specific number, but for the enzymes of unknown function, it's like a very...
Yeah, because I think the proteins was really, if we go into database and there is some hint in the annotation that it's still uncertain, okay, we're like, okay, we call it the protein of unknown function. The enzymes, as I told you, it's based on sequence and there is a lot of, it can still, there can still be parts of the sequences conserved, but still might do something completely different, right? So it's just, we have used different strategies to try to estimate this, but it's really not a precise science, so, yes, Alex? Yeah, so many of the unknowns are conserved. That's also quite interesting. Which even further, tells us we should not just leave it like this, even though it would be more. Okay, so this is just now to show you a bit in this map that we come here to a bit of a paradox if we accept that indeed there are still also among these enzymes of unknown function a lot of metabolic enzymes that, if we say we have this map, they are in our textbooks, they are, we know where to go, they are complete, but then you do this, look at these databases and you find that there are still so many enzymes that seem to catalyze metabolic reactions, but that are not in this map. And so it just tells us, either, within this map there are intermediates that we have not figured out. There may be total pathways that are missing, and also when we are using this technique that I'm saying, like metabolomics, so where we try to measure as many of these metabolites that we have at a given time point in a cell, or we can also do it in blood, for example. Actually, we have many peaks that we still don't know how to connect to this network, which is another indication, without going to this exercise of the protein databases, that we are missing something. And so then you start thinking about possible enzyme functions, and I can then come up with a list of at least where should we start thinking about this. And I think when you look then into the biochemistry literature, you can see where are the domains, where are the where we still have a lot of new functions that are discovered. And I would say, this kind of summarizes this more or less. So indeed, proteomics also shows us that there are a lot of post-translational modifications. So one that you certainly know are phosphorylations. So you find these phosphate groups on serines, threonines, tyrosines of proteins that modify their functions of the proteins. But there are many other modifications that you can do on proteins, and some modulate more or less the function of these proteins. So and it's enzymes that catalyze those modifications. So I would say, there is things to find also on RNAs. So our RNA molecules are kind of decorated with modifications of small molecules that, again, they have to be put on there, bonds have to be created, and they are catalyzed by enzymes. And there is still a lot of things that are elucidated there as well. Some of these, what we think are catalytic proteins, because they have an NAD binding domain or an ATP binding domain, or something that seems to bind a metal cofactor. Some of them, they might have also lost their catalytic function, and in the meantime, they have become a regulatory protein. And I will show you an example of this. And then you can make this distinction between primary and secondary metabolism. So I usually say that the primary is what we really need to run as reactions to stay alive. whereas secondary is more optional. I mean, you know, so microbes, for example, do it a lot to fight the other species that is in the medium. So they form antibiotics, and that's like a secondary. They don't need it to per se survive, but they, yes, in their environment, they need it to fight against others. So secondary metabolism is actually typically not done a lot or at all by humans, and also not so much by yeast. And so because we have found these many enzymes of unknown function in also still in humans and yeast, tells us, I mean, you could say, you know, it's all doing secondary metabolism, because there are lots of weird compounds that we, in the phytoplankton probably, that we have no ideas yet how they are done enzymatically. But because we see it in organisms that don't do much secondary metabolism, it seems to be still also some of this connected to primary metabolism. And then that brings us, and there are still gaps, but I would say in this map that we know, there are really not many gaps anymore. So that's why something tempting is that there might be also in metabolism this things going wrong. I mean, biology not being perfect, side products being formed, and the necessity of enzymes that actually repair things that are going wrong in metabolism. And that is something that I will elaborate on much further. And before I'm going into that category, I wanted to make still you know, if you are now in this position and you say, okay, now I want to find out, I have one protein, I don't know, I like it for a certain reason, I want to find out what its function is. And I think it's an enzyme. Also, what you need to keep in mind, and what becomes more and more clear, is that many enzymes have actually not only one function. And that can be more subtle differences. So it can be because of what I told you before, in different organs, the same enzyme will be regulated in a different way. So you can say it's more than one function, but that's just a property that is different, and that can come about by producing isoforms from the same gene by alternative splicing. But also many enzymes have actually several catalytic domains, so they execute more than, you know, if you have found one, well, you are not at the end of your project, but because there are still 2, 3 other catalytic sites that you need to figure out what reactions they are catalyzing. Then there can be one enzyme can recognize several different substrates and then also catalyze different reactions on them. We have this notion of moonlighting, where some enzymes have, next to being, you know, catalyzing this isomerization in glycolysis, they do something completely different that has nothing to do with their catalytic function. And then this example of, you know, some of the enzymes that actually have lost their catalytic activity have become regulators. And so I'm going through that with a few examples to just illustrate it. So this alternative splicing, which brings us away from this dogma, one gene, one protein, or in our case now, one gene, one enzyme, that's actually often is one gene and several enzymes or several proteins. And that comes about by alternative splicing. So when you have your messenger RNA with the exons and introns, there is, you know, like the machinery has often a choice between using one exon or actually like skipping it or like bringing these exons together in the coding sequence in different ways. And so you can form different variants of the same enzyme. And that can then make that they are differentially regulated. Like, for example, liver but not muscle phosphofructokinase 2, which is an important regulator of glycolysis, is phosphorylated by, so the regulation is different of these same glycolytic enzymes in different organs because of such a difference in sequence. Also, it can make then that you have one same enzyme that have different N-terminal extremities, so then it can be brought to different subcellular compartments. And you have like these differences in sequence that can actually make that one form of the enzyme recognizes substrate S and another form recognizes another substrate. So this multiple catalytic domains, they mean simply that you bring together several domains and in several of the same protein sequence, but then in one part of the enzyme you catalyze, you know, one reaction, in another part another one, and yet another in the other part yet another one. And that, you know, brings sometimes this notion of substrate channeling. I don't want to go into this, but some believe that this is needed to make catalysis and such sequence of reactions more efficient. And I mean, we like this in our enzyme function identification domain because, you know, if you have figured out at least the function of one of these domains, it can become easier to figure out the functions of the others because typically these functions are connected. And, you know, bringing them together in one protein helps you to more rationally, in a way, regulate them, because if they are needed, they will then all be expressed together. If there is a regulator, it will be regulating all these domains together. And so the multiple substrate thing is actually important to connect to my theme of repair, because the notion that some enzymes recognize many different, chemically different substrates is not something new, because there are really entire family of enzymes, at least in mammals, for example, that recognize indeed a big complexity of molecules, which are typically these enzymes that act on xenobiotics, so molecules that we do not produce ourselves, but for example drugs or other things that come in from the environment. And those enzymes, the drugs, as you know, they are metabolized also in our organism, and then we secrete them under a more hydrophobic form that they came in. And those enzymes, they have this property that they can recognize chemically quite divergent, quite different compounds. So that's, I would say, is a broad specificity simply. They act on substrate 1, on substrate 2, but with similar activities, with a similar velocity. But then there is this also, and I would say this is true for like many enzymes, that there is no strict specificity. So you often show this in textbooks, this key on a lock notion, that like really one substrate, specifically just that one fits into the catalytic site and cannot do anything else. This is really, I mean, this is just, it's actually relatively true because they are remarkably specific. But, you know, if you are an enzymologist and you express an enzyme, you work on it in vitro, and you give it something that looks alike, it will also catalyze a reaction on this, maybe 100 times slower or more than 1,000,000 times slower, but this can still, you know, like have a meaning, have a consequence for the organism, as I will show you. Moonlighting, so this is easier. So it's basically, although it's breaking sometimes our mind, because it's that some enzymes, they have a well-established catalytic function, but then you knock them out and they have completely different effects than what you would expect from just not this reaction being there anymore. And that's because they also, for example, phosphoglucose isomerase, a glycolytic enzyme that incorporates glucose 6-phosphate and fructose 6-phosphate. It's also acting as a neurotrophic factor, so it's secreted, and then it's helping neurons to be maintained or grow. And by tumors, it's secreted as a cytokine and angiogenic factor, so completely unrelated to its catalytic function, and also certainly due to other parts of the protein. So it's not involving this catalytic site. And then a last one, so I think the example is nice because phosphofructokinase, an important enzyme in glycolysis that Matthias has shown, this one is now a dimer of two proteins, that where the one domain is still indeed converting fructose 6-phosphate to fructose 1,6-bisphosphate. But the other domain, it's a homologous to this one, so sequence-wise it looks very similar, but it has lost its catalytic activity, but it's still binding something that looks very similar to its actual substrate, because it has evolved from this protein actually, and now it's taking this property to regulate. It binds it modifies its structure, its configuration, and it's actually then having an effect on how fast this reaction is catalyzed here. Okay, so this complexity, of course, makes it even more difficult to, in a straightforward manner, link a function to a protein of unknown, or an enzyme of unknown function. But it's good to be aware of it. Then coming to this notion, yes. Sorry. Yeah. Yeah. Yes. You still should remember it like this. I mean, it's I'm talking here about, you know, the weird things maybe in metabolism. So I'm giving more, but it's you know, it's, of course they are mainly catalyzing these reactions. Some are not mainly. Some are like catalyzing this and having really other important functions. But yes, I mean, enzymes are catalysts which facilitate or accelerate chemical reactions in cells. So that's still their main function. So then, yeah, indeed. Some are will be annotated because of their sequence. They look very much like indeed catalyzing, you know, similar to one that is catalyzing a reaction. But you can have the surprise that actually, in a certain species, has evolved to not anymore doing this function, but the protein is used for executing another function. Yes. But overall, yes, enzymes are. So certainly, this notion of biology not being perfect and repair being needed in biology is. Some of you might be very well aware of it because of what's going on DNA. So in DNA, you know that it's completely essential that we maintain the mutation rate low, that every replication we avoid errors, and you know we very faithfully transmit to the next cell the genetic information that we had in the mother cell. And even very small errors there can be super detrimental. So you know, everything has been made to decrease this mutation rate as much as possible. And one of the mechanisms is that the polymerases that actually form the new chains of DNA during replication, they have among the domains that they need for executing this function. So integrating the new nucleotide complementary to the mother strand. They also have this green exodomain, which actually recognizes if should there be an error happening, and should there be a non-complementary base integrated. This exonuclease nuclease domain will feel this and will actually then hydrolyze the bond and give the enzyme the chance to integrate the correct one. So that's what we call the proofreading in function in DNA, and in a way it's really due to the fact that even though polymerase is almost perfect in always putting the right nucleotide in here when it's synthesizing the new strand. Sometimes it's making a mistake, but then there's a backup because there is this exonuclease domain that will recognize this. Similarly, in protein translation, so there what you put your amino acids that are like forming your polypeptides, which are the proteins. You have to activate them and then put them on these tRNAs that are then going into the ribosome for building up the protein. There is a similar mechanism. So in these tRNA synthetase enzymes, there are domains that will recognize if a wrong amino acid has been charged on a certain tRNA, will hydrolyze it and will give, you know, the chance again to do the right thing. So, and I think this can help you understand like this notion in that we are pretty sure now is also an important component of primary metabolism, where the enzymes that are catalyzing these intermediate reactions in our primary metabolism, they have evolved to be quite remarkably specific, but still they are not perfect in the sense of specificity, and they will catalyze side reactions, sometimes at a much slower pace. But if the product of that side reaction somehow, for example, could imagine has toxic effects, then the way that nature seems to have found to counteract this lack of being perfect is to have also evolved enzymes that actually recognize these weird minor side products and reconvert them into precursors that are actually useful to to metabolism, for example, or are actually at least not toxic. And this, I think, we have published a review on this where we had already several examples in 2013. That was like when we came up with this concept and thought that it's actually maybe much more pervasive across metabolism than the few examples that we knew by then. But it's more and more of such enzymes have been found, so the side reactions and also repair enzymes. And so it helps us actually decreasing slowly this list that I showed you of enzymes of unknown function. I'm not saying that all of these enzymes are repair enzymes, but a fraction of them are actually repair enzymes. And for a long time we neglected them because, of course, it's more important to first figure out the main roads and the big highways. And also technically, you know, these things are, because they are actually repaired, this will be typically super low concentration. You will, in a normal cell, you will not even measure it, even if you have very fancy metabolomics techniques. If you are not looking for it, you will not find it, basically. And so the concept had to almost come up before people were really looking for that. And I find it always really interesting to show how this concept was born, because, you know, many people working more in fundamental research and so on, they don't want to hear about disease, and it's, you know, I just don't do it for the disease and for the drugs and so. But actually, disease can teach us a lot. Notably in metabolism, I would say that this map that I said is not complete, but you saw it's quite impressive. A lot of the knowledge in this map comes from metabolic diseases, where something in this map, some reaction is not working because there is a small mutation in the gene and the reaction cannot happen. And at that point, things appear in your blood, in your urine, you know, that we can now measure because just now the flux is blocked and these things accumulate, although normally they are very low concentration, and we see them, and before we didn't even know of their existence. So this has taught us a lot about metabolism and to elucidate many of these pathways that we now know. This, you know, unfortunately, luckily they are rare, these diseases, these inherited metabolic diseases, because then if you have them, they have very severe consequences, often childhood diseases with very early death, etc. The thing is that, I mean, they by of course looking at these patients, trying to understand what's going wrong, we figured out a lot about metabolism as well. And now sometimes we can actually even treat them because we know a lot about this metabolism. So I will show you some of this. And so what I want to bring to you now is how it came about, this concept of metabolite repair. It was in the context of an inborn error or an inherited metabolic disease. There were patients in the 1980s already known that accumulated in their urine and their blood, in their extracellular fluids, a lot of this decarboxylic acid, L2-hydroxyglutarate. And that remained a mystery for a long time because it is not an intermediate. It's not any dot of this point, of this map that I showed you. It doesn't fit into our pathways. So we didn't know how it was produced, we didn't know how it was turned over, and for a long time, I mean for several years, we just had no clue why and also we know that it's not abundant in nutrition, so it's also not something that they take in and then accumulate because they don't have the possibility to degrade it. But I mean, through genetic and biochemical approaches, it was then figured out that indeed in our arsenal of proteins, of enzymes, there is one enzyme that is actually not linked to NAD but FAD, that indeed recognizes very well this strange decarboxylic acid and converts it to something that is extremely close in chemical structure, but that is now, I mean, a very well-known intermediate of our metabolism. So we have converted something that appears to be toxic, and we don't know, you know, how it is formed, into something that can be very well used. It's an intermediate of the TCA cycle. And so it's not far away, right? It's just an oxidation reaction, but there is this enzyme that indeed recognizes this weird decarboxylic acid, and that's why in normal cells you don't find it, because there is this enzyme. But these patients, they had a mutation in the gene that encodes this enzyme, and so they started to accumulate a lot of this, and it has neurotoxic effects for reasons that are not fully elucidated yet, but that brought the problems. So even if we had understood this, there was still this question mark: how is this hydroxyglutarate actually formed? So we know now it can indeed in the cells be converted, and that's why normal people don't have a problem with it. But how is it actually formed? And there was a lot of classical approaches in terms of like trying to find an activity that's actually forming this hydroxyglutarate in liver extracts, for example, and then trying to purify that out. And in the end, what was found is that actually this reaction can be catalyzed in a very slow manner by an enzyme that is another enzyme of the TCA cycle, and that in this TCA cycle is interconverting oxaloacetate and malate. And if you look at the structure, so oxaloacetate, one of its substrates, is really quite close to this alpha-ketoglutarate. It just has one CH2 group more in the chain. So on the one hand, you can see remarkable specificity because it's quite close in structure, but here the Km is 0.012 millimolar and the Vmax 220 units per milligram. When you put one carbon more here, the Km becomes 8 millimolar. So to be at half the maximal velocity, you need, more than 100 times, 1,000 times more than for this molecule, and it has a much lower maximal velocity. So in efficiency, more than 1 million times less efficient on that than on this. So actually, you can say it's highly specific. But at the beginning also we were a bit puzzled. I mean, is this, so we could measure it in vitro, but is this really going to bring about all that trouble if you don't have this enzyme? If you don't have this enzyme, this slow activity, can it bring about all these problems and even like lead to a deadly disease? And so now that we had the kinetics of that side reaction, so here's the main reaction, here is the side reaction that you can of course determine. Once it's measurable, you can determine the kinetic properties. We also could, I say we because it was actually in the lab where I did my master's thesis, but it was I was not really involved in that work. So so we could determine how much of that enzymatic activity, for example, we had per gram of liver. And with that, we could then calculate actually, for instance, what amount, so with that, with the kinetic properties, with the maximal activity, here is basically the Michaelis-Menten equation that is used to just calculate the activity of the enzyme at the concentration of alpha-ketoglutarate that you find in the mitochondria, where it is really abundant for metabolite. 1.56 millimolar is, for an intermediate, it's quite abundant. And then, like converting it so that to bring it to the 1.5 kilo of a liver, we could calculate that you form by this very poor activity 7 millimole of this decarboxylic acid per day. If you take into account that kidney, muscles, etc., also do this, so you can easily imagine that this is explaining the 26 millimole per day that is found in the urine of the patients. And so, yes, I mean the fact here that you have an enzyme that is in the mitochondria very abundant, that you have its secondary substrate or its side reaction substrate that is also highly concentrated in this compartment where it is highly abundant, makes that with this small side activity you can form enough that it's really perturbing metabolism if you do not have an enzyme to sense it actually, because the Km of this enzyme for this weird byproduct is quite low, meaning that its affinity is high. And so as soon as it is produced, it will be converted here. And you know, you might think, okay, this is just like 2 times the same, just in the other direction. It is not, because this enzyme is NAD bound and this enzyme is FAD bound. And I think the thermodynamics of this is quite different. So yes, so that's the example of malate dehydrogenase producing this L2HG, posing problems, but the repair enzyme is there to bring it back to an intermediate of the TCA cycle. So that's like a side reaction that's going on here in the TCA cycle. There is another enzyme in the TCA cycle, isocitrate dehydrogenase, that is actually converting alpha-ketoglutarate in another side reaction to just a stereoisomer of this, where the hydroxyl group is on the other side. There is another enzyme that now recognizes this enantiomer of 2-hydroxyglutarate, and again, if it's absent, it leads to severe disease. You can have another mechanism where certain mutations of this isocitrate dehydrogenase are found in certain cancers, and that actually brought a lot of attention to it all of a sudden to this whole thing. In this case, this mutation makes that this side reaction becomes huge. I mean, it starts to do that like crazy. And then actually the capacity of this repair enzyme is completely overwhelmed, and you however accumulate this, although you have this repair reaction. And so I want to talk about one more example of such damage and repair, and I think it's a nice example because it really affects, it shows you, already the first example showed you that it can affect really very central intermediates of our metabolism, but also this example is actually shows you that also very central cofactors can become damaged within our cells, and that there are actually important repair mechanisms to avoid that this causes problems in the cell. So coming back to the textbook knowledge, just to illustrate again, NAD is this cofactor of many, many enzymes, and notably in our central pathways here of glycolysis and TCA cycle, where one of the, let's say, byproducts of oxidizing glucose is to form NADH. Also in the TCA cycle, you have a number of steps that will reduce NAD into NADH, and all of this NADH in the end is, or some of it can be shuttled into the electron transfer chain, where then you have the ATP synthase in the end that is forming our energy. Other than this, there are thousands of enzymes that use NAD or NADP as a cofactor. And here is the structure of this cofactor. So it's a dinucleotide, and the important part here is on this part, this moiety of the NAD, you have this important cycle, the nicotinamide cycle, where you can indeed have, actually it's on the 4th carbon here, the hydrogen that is exchanged in these reactions. And it works so well for these oxidoreduction reactions because when you take off the hydrogen, then you have a positive charge actually, but it can be stabilized by these conjugated bonds here. So that's how it's used as an oxidoreduction central cofactor. It's also, and I alluded to this earlier, actually that's a much more recent knowledge. So in general, NAD, all these oxidoreduction reaction, that's something that has been figured out since a while. But more recently, it has become clear that very important metabolic regulators like the sirtuins, poly-ADP-ribose polymerases, they use NAD not as a oxidoredox cofactor, but really as they need NAD as a molecule in their reactions. For example, the poly-ADP-ribose polymerases, they recognize damaged sites in our DNA, and then they start building like polymers of ADP ribose, which is this part, this part of NAD. They polymerize this moiety and kind of like flag the places for attracting the DNA repair machinery. So there, it's not like NAD converts to NADH and back, but it's really a part of the moiety of NAD that is used. And this is quantitatively also quite a lot of the NAD that is needed by these reactions here. And so it is obvious, like with all of these functions, that you need a very well-tuned pathways to form this NAD and to maintain its level. So as for glucose homeostasis, you need to maintain an NAD homeostasis. And we have actually redundancy in this system, because you can form NAD from an amino acid, tryptophan, at least in mammals, it's done in this way. Via multi-step pathway here from tryptophan that you successively convert into into NAD. Then you have this that one of our B vitamins, vitamin B3, is actually a precursor for NAD. So it's exactly this moiety here, vitamin B3. It's a bit of a Sammelbegriff, so it's there are more forms. I think vitamin B3 also can be the nicotinic acid, so without this amine group here. In any case, both of them can be, so nicotinamide or nicotinic acid can be via different pathways put together, like with others, to form this NAD. And then there are, so this is de novo synthesis, but you can also recycle. So once these big consumers of NAD act on it to break it down, you still maintain this important moiety here, NAM or nicotinamide, and then there are enzymes that can bring that back into NAD. So you recover you do not deplete your cofactor pool. And also, like, then you can also imagine that dysregulation in any of these pathways can lead to disease, because it's such a central thing. And also, as with any of the vitamins, if you do not have enough, although you have these pathways of de novo or salvage pathways, you still need a constant supply of B3 from the nutrition to really maintain NAD. Because if you don't have enough, and that doesn't happen much often anymore in our societies now, but if you don't have this vitamin B3, you develop a disease that is called pellagra. It's the 4Ds, dermatitis, diarrhea, dementia, and ultimately death, if you do not have enough B3 in your nutrition. And there are other deficiencies, also primary deficiencies in these pathways, that can lead to NAD depletion, and then that leads to disease. And so this has been figured out for a long time, but now with our notion of these damage and repair reactions, actually we can add a pathway here, so short pathway, but it's clear now that the reduced form of NAD, NADH, is constantly being damaged in our cells by hydration reaction. Transforming it into a molecule that is not acting as a cofactor, cannot act as a cofactor anymore, that then converts to a cyclic form that we cannot do, that is really lost to the pool of NAD. But we do have a machinery to repair those hydrated forms of NAD. And actually, it does go back very far in the biochemical literature, because already Edwin Krebs and his team in the 1950s figured out indeed that this glycolytic enzyme, GAPDH, in addition to catalyzing its glycolytic reaction, if they left the enzyme with in the presence of the cofactor for sufficient time in the spectrophotometer, so you know that's what enzymologists do. This type of reaction they measure by putting the absorbs at measurement at 340, because this one absorbs at 340 and this one not, and so we can know that the reaction is going on. So at the beginning you see a linear and then like saturated increase of this absorbance, but then all of a sudden over time it is going down. So something else is happening with this NADH. And even in that time, and then in the 70s, it was figured out that this something else is actually a hydrated form of NADH. So what this enzyme seems to do as a minor side activity, and I mean, I think even structurally and so on, it's not yet well figured out how that happens in the catalytic side. But it's basically adding a water molecule onto this double bond, which then leads to a hydroxylated form of the NADH. You see that we lose this double bond, which is detrimental to the cofactor activity, because now if you give your hydrogen here, you cannot stabilize the positive charge anymore. So it does not act as a cofactor anymore. This hydroxyl group can be in an R or S configuration, so you can form two types of this NADHX, as it was called before its structure was elucidated. It's not a cofactor, and it actually, because it's similar to NADH still structurally, it interferes with metabolism because it can inhibit some of the many NAD-dependent enzymes. And so we thought this is really a nice example of something where we know already there's a side reaction. There seems to be a side reaction happening. If this is really going on in the cell, there should be an enzymatic mechanism to repair this. And it was actually relatively easy because of what I told you. So this is something we can easily measure in a spectrophotometer, and once it's hydrated, it's also gone. This absorbs at 340, so we can easily set up an enzymatic assay. We took our favorite model organism, which is budding yeast, to find this activity even in a complex extract. Was no problem. And then we purified it, and with a biochemical approach, we figured out what the sequence is that carries this activity. And so then proteomics guys came back with a number of candidate sequences that could underlie this enzymatic activity, and it was annotated indeed in the time as uncharacterized protein. I think also like carbohydrate domain containing protein, something like this. So now that we had the sequence, we had one strength, which is to figure out, I mean, is it only in yeast or is it conserved? And actually it's super conserved. You find it in every living species, maybe not in, I mean, there were some exceptions like some parasites, if I remember correctly, but otherwise it's super conserved. And interestingly, in E. coli, it's actually fused to another domain of unknown function at the time. And as I told you, that actually was an interesting hint for us, because it told us maybe it's also functionally related to this NADHX repair. And then it turned out to be indeed just an epimerase that is interconverting these two forms, the R-NADHX and the S-NADHX. And this is important because the one that is taking off really the hydroxyl group, the water molecule, is stereospecific for the S form. And so you need this whole system to take care of all the forms. And this enzyme also is conserved in all of the other species that have this. But in humans, for example, they are not fused together, so it's not needed. But it was for us an interesting fusion here to hint us towards the additional function. So that's another example now of metabolite damage by a glycolytic enzyme that converts NADH into non-functional cofactors. But it can also come about through, for example, high temperature, acidic conditions, and so on. That's how we produce it in the lab. So this can also go spontaneously. It's okay. I mean, we can deal with it because of this repair enzymes. Maybe just as a side note here, this is an example of an enzyme, this NADHX repairing enzyme. It can have, it can be spliced differently, and so you can target it to the mitochondria. Another form is targeted to the ER, another form is retained in the, so this is a cell, this is the nucleus. These dots here are the mitochondria, it's indicated by this mitochondrial marker. This is an endoplasmic reticule marker, so if it is this form, the enzyme is in this compartment. If it doesn't have these flags that bring you to a certain compartment, it stays completely in the cytoplasm. So it shows that it's not only like distributed across all species, it's also really brought to all compartments of the cell, because NAD is just also everywhere. It's enriched in the mitochondria because NAD and NADH especially is really concentrated in the mitochondria. So there you probably need it a lot. So then we have done everything in vitro. So we were with the isolated enzyme, and then biology journals also say, oh, this is just in vitro, you have to show that this happens really in the cell, and it's true. And so when we knock out this new enzyme in yeast, for example, indeed we see high accumulation of this weird NADH derivatives that had never been measured before in a cell extract. And actually also you see that it messes then up, at least in such batch cultures, the NAD homeostasis, because the normal NAD levels are going down. I think I don't go into that, because then we wanted to know like further, and here is again this notion of transcriptomics and metabolomics, but Alex will explain this more. And now of course our interest turned to, okay, when this enzyme is not there, what happens with the cell? Actually in yeast it's not detrimental, at least under the cultivation conditions that we use. They actually grow fine. But then joins a bit the question that was asked before, like can you know that it really has a function? So we know that it really has a function, but the yeast still do fine without it. However, we could see signatures in the metabolic profile and in the transcriptomic profile that showed us where in metabolism is going to mess up things mainly. In this case, it was actually in serine production. So we could show all that. And then actually only after we did this fundamental work, and starting from yeast and even from an isolated enzyme only, several years later, there were doctors that contacted us because they had kids that seemed to have like a mitochondrial disease. I mean, all the symptoms indicated to them that it might be a mitochondrial deficiency, because they had these metabolic perturbations and severe neurological episodes and so on. But the exome sequencing, because all of the usual suspects were fine, the genes were fine, mitochondrial genes, etc. But the exome so the sequencing of the genomes of these kids brought them to the NAXI and the NAXD enzymes. Those were mutated in these kids, and whereas yeast can cope with this deficiency, more multicellular, high complex organism cannot, and especially the brain is suffering a lot. However, also the skin was severely affected, which certainly brings us back a bit to this pellagra-like symptoms. So I think you have both the problem that you have a shortage in your normal cofactor, but also the derivative is going to interfere with other reactions in metabolism. So we modeled this also in human cells and found again like this major perturbation in serine metabolism, which will maybe be helpful in treating these patients, because what we know, I will not go into this, but if you give those patients vitamin B3, so this precursor of NAD that you indeed would expect to deplete in these repair deficient individuals, you can see that you very nicely can act on, for example, the skin lesions. The doctors also tell us, but I think we need longer years now of seeing whether over time this indeed stabilizes, but apparently neurological symptoms are stabilized. Our worry with this treatment is that when you look, for example, we have a zebrafish that is deficient in the repair system, and they die spontaneously even without a trigger. Actually, I should say these patients they are born fine, and the symptoms start upon a trigger like fever or a hit against the head, some kind of stress that seems then to really bring about the whole symptomatology. The zebrafish actually, if they have maybe in zebrafish we have even a spontaneous phenotype developing because we fully knock out the enzyme, and I think in humans there is always, even in these patients, a little bit of function that keeps them over water, and then but once there is like an insult, then they cannot, then they decompensate. But so the zebrafish are dying compared to wild type when they don't have the repair enzyme. And you can see if we give them B3 in their water, there is a substantial increase in survival, but they will end up dying nevertheless, prematurely. And it's probably because we initially do something good by by giving them more NAD so more precursor, more NAD, more of the healthy cofactor, but more of the healthy cofactor means also more of the toxic byproduct, and this eventually is probably then going to be having adverse effects. So I think I skipped the. There are other examples, of course. I mean, one other one actually that is nice to tell, because it has also already led, since we understand the damage reaction and the repair reaction, we also now understand an inherited neutropenia that is due as well to an absence of a repair phosphatase in this case, which then leads to the accumulation of a sugar analog that is phosphorylated, that inhibits glycolysis, and that turns out to be especially detrimental to our neutrophils, so the white blood cells that are fighting infections. The neutrophils of those patients are dying off, because those are very dependent on doing glycolysis, pentose phosphate pathway. Also the other cells, but they seem to be even more affected than the others by this. And so their neutrophils are dying, and they have recurrent infections all the time. But now that we figured out the mechanism of what molecularly is behind this, we can actually we can quite effectively treat them. Good. So I want to come to the conclusion here. So just showing again these central pathways of glycolysis, TCA cycle, and here a little piece of the pentose phosphate pathway. And in gray, I show enzymes for which we know already now that they catalyze definitely measurable side reactions with precursors that are present in the cell, because I mean, if they are not present in the cell, it's not a problem, but if they are, then they can also form these side products that could then interfere with metabolism and other functions. In red here, I'm showing the side products that are formed by these side reactions along these pathways, and we have talked about some of them: NADHX, D2-hydroxyglutarate, L2-hydroxyglutarate. So in yellow, I think, no, here I show in green now the repair enzymes that have been found to reconvert these red side products into normal intermediates of metabolism, or even degrade them just so that they don't interfere with metabolism. And in yellow are the ones that we have spoken about, at least briefly. And then in here in pink or purple, I'm showing those that have been already connected also now to human diseases. So showing what I said as in the title. I think you can believe now maybe that repair damage is also happening in metabolism damage. Of course, it's a human word that we use, but what I mean is that non-wanted reactions can happen, and that you need to find a system to deal with these non-wanted reactions, and what we use are these metabolite repair enzymes that convert these side products back into useful intermediates. And I say, it's also damage and repair is also occurring in metabolism, not only in DNA, not only in protein translation. And it's essential because, as you saw, if you take away one of these repair systems, it's not compatible with life, at least in humans. And so, yes, the conclusions, if you want to have a brief summary of the properties of these enzymes, they metabolize non-canonical, potentially toxic metabolites. Also, cofactors can get damaged, as I told you, with NAD. They are formed endogenously by enzymatic side reactions, so we are not here talking about the xenobiotics that are coming in, or enzymatic side reactions, or even unwanted chemical reactions. Sometimes they are not catalyzed by enzymes. And they may account for still many of the putative or enzymes of unknown functions that are in our encoded by our sequence genomes. Why am I thinking this? Because there are many more enzymes with side activities than the ones for which we have now matched the repair activity. We have many peaks in metabolomics analysis that are corresponding to metabolites that we just don't know yet where to fit them into this network. they are important enzymes to look for. I think you will agree just for our fundamental understanding of metabolism, but also because they help us understand detrimental diseases, and by understanding those diseases we can actually do something about them, sometimes at least. In metabolic engineering, they start also using this concept to optimize their metabolic engineering systems, because as you could imagine, it's annoying if you just like form these side products, and they overexpress often metabolite enzymes in organisms where they are not usually expressed. So if you overexpress an enzyme, of course the side activity becomes also maybe more important, and if that then interferes with their engineered metabolic network, it's certainly not optimal. So they will then also put in the repair enzyme to avoid this. And with that, I think I'm out of time, and thanks for your attention, and of course if there are questions.